Ever get that feeling? Like you're drowning in information.
Yeah.
Could be for work, a new hobby, anything really. But you just want the core, the absolute essentials,
Hmm.
and you don't have time to wade through a mountain of articles and books.
Oh, yeah, I totally get that. We're all busy these days, right? Who's got the time?
Right. So that's where we come in. This is the deep dive, your shortcut to understanding complex topics.
We're your guides, ready to cut through the noise and get you straight to the juicy bits of knowledge.
Exactly. Today, we're diving deep into a fascinating area, autonomic pharmacology, which basically means how medications interact with your autonomic nervous system. We've got a stack of material from a pharmacology textbook, chapters 6, 7, 8, and 9, plus some relevant sections from section 2 on autonomic drugs, all about the ins and outs of the ANS and the drugs that can influence it.
We're talking diagrams, tables, case studies, the works. Our mission today is to distill the most critical insights from all this. We want to make it clear, engaging, and help you grasp how medications can affect this crucial part of your body.
And this is for everyone, not just medical professionals. Whether you're trying to understand a medication you're taking or you're just curious about how your body works, this deep dive is for you. Let's get started by talking about the star of the show, the autonomic nervous system itself.
All right, so the autonomic nervous system, or ANS for short, is constantly working behind the scenes, keeping your body running smoothly. It controls all those vital functions you don't have to think about, like your heart beating, your digestion, breathing, sweating, and even things like pupil dilation. We're talking truly automatic stuff here.
That's a lot of responsibility. How is the ANS organized to handle all that?
Well, it's divided into two main branches that often have opposing effects: the sympathetic nervous system and the parasympathetic nervous system.
Sympathetic and parasympathetic.
Yeah.
Okay, so those names sound pretty different, but how can I easily remember what each branch does?
Think of it like this. The sympathetic nervous system is your gas pedal, your fight or flight system. It kicks in when you're stressed, in danger, or excited. It gets your heart racing, your breathing faster, your blood pumping, ready for action.
So it revs you up. Got it. So then the parasympathetic nervous system must be the brakes, right?
Exactly. The parasympathetic nervous system is your rest and digest system. It slows things down, helps you conserve energy, and focuses on things like digestion, lowering your heart rate, and calming you down.
Okay, so gas pedal for sympathetic, brakes for parasympathetic. That's a really helpful analogy. Now, where exactly is all this happening in the body?
So the ANS involves both your central nervous system, which is your brain and spinal cord, and your peripheral nervous system, all the nerves that extend out from the CNS to the rest of your body. These autonomic nerves connect to your organs and tissues, allowing them to receive those gas pedal or brake signals.
And you mentioned earlier that the ANS does a lot more than just the obvious stuff. What are some of the other roles it plays?
It's involved in regulating your immune responses, for example. It can even influence certain processes within the central nervous system itself, impacting things like mood and sleep. And we can't forget about the enteric nervous system, often called the brain in your gut.
The brain in the gut. Okay, that sounds super interesting. What's that all about?
It's this incredibly complex network of neurons located within the walls of your digestive system. What's amazing is that it can operate somewhat independently, meaning it can control many aspects of digestion without direct input from the brain. However, it's still influenced by the sympathetic and parasympathetic nervous systems, receiving signals from both of those branches.
Wow, so we have a whole other brain down there taking care of business. What kind of things does this gut brain do?
It does a lot. Just like your brain, the ENS has sensory neurons to detect things like the presence of food or changes in pH. It has interneurons to process that information and make decisions. And it has motor neurons to control things like muscle contractions to move food along, and the secretion of digestive juices and enzymes.
It's a whole system down there. Fascinating. But how does it communicate and coordinate all these activities?
Well, it uses an array of neurotransmitters, those chemical messengers that we also find in the brain, like acetylcholine, norepinephrine, serotonin, dopamine, even things like VIP, substance P, and enkephalins, which are involved in pain perception and modulation.
Wait, so those same brain chemicals are also at play in our gut?
They sure are. And just to give you a sense of their roles, acetylcholine in the ENS often has an excitatory effect, promoting muscle movement and secretion, while norepinephrine tends to have an inhibitory effect, generally slowing things down. Of course, it's a very complex system with lots of nuanced interactions. Now, to understand how drugs can influence all of this, we need to talk about how these nerve cells talk to each other in the first place.
Okay, let's get down to the cellular level. How do these autonomic nerves actually communicate?
The communication happens at synapses, which are those tiny gaps between nerve cells or between a nerve cell and an effector cell, like a muscle or gland. The main form of communication here is chemical. When an electrical signal, an impulse, travels down a nerve cell and reaches the synapse, it triggers the release of neurotransmitters from the presynaptic neuron, the nerve cell sending the signal.
So these neurotransmitters are like the words being spoken in a conversation.
Perfect analogy. These neurotransmitters diffuse across that tiny gap and bind to specific receptors on the postsynaptic neuron, the receiving nerve cell, or on the effector cell. This binding then causes a response in the receiving cell, either excitation or inhibition.
And it's the type of receptor that determines whether the response is excitatory or inhibitory, right?
Precisely. It's like a lock and key. The neurotransmitter is the key, and the receptor is the lock. Only the right key can fit and unlock a specific response. Now, drugs can step into this conversation and mess with things. They might mimic neurotransmitters, acting like a fake key to activate a receptor. Or they might block neurotransmitters, preventing the real key from fitting into the lock. They can also affect the release of neurotransmitters or how quickly they get broken down.
So medications are essentially hacking into these cellular conversations. Fascinating. Now, who are the main players in this neurotransmitter world? Which ones should we be focusing on?
There are many, but the two most important ones in the autonomic nervous system are acetylcholine, or AHE, and norepinephrine, or NE. We shouldn't forget epinephrine either, also known as adrenaline, especially when it's released from the adrenal glands as a hormone, though it can act as a neurotransmitter in some brain areas too.
Acetylcholine, norepinephrine, and epinephrine. Got it. Let's break down each one, starting with acetylcholine. Where does it act, and what are its main effects?
Acetylcholine is a busy bee in the ANS. It's the primary neurotransmitter in all autonomic ganglia, those relay stations where signals are passed between neurons in both the sympathetic and parasympathetic pathways. It's also the main neurotransmitter at parasympathetic postganglionic junctions, where the parasympathetic nerve fibers meet their target organs. Interestingly, it's also used at some sympathetic postganglionic junctions, specifically those that control your sweat glands. And remember the enteric nervous system. Acetylcholine is key there too.
Wow, it's involved in a lot. How does it actually carry out its functions? What's the process?
It all starts with its creation. Acetylcholine is synthesized inside the nerve terminal from choline and acetyl-CoA, a molecule involved in cellular energy production. An enzyme called choline acetyltransferase, CHACR for short, does the building work. Then, this newly synthesized ASHE is packed into tiny sacs called vesicles, and a protein called the vesicular acetylcholine transporter, or VAT, helps with that packing. When a nerve impulse comes along, these vesicles fuse with the presynaptic membrane and release acetylcholine into the synaptic cleft.
So it's like a carefully orchestrated release. What happens once acetylcholine is out in the synaptic cleft?
It quickly diffuses across the gap and binds to those specific receptors we were talking about, cholinergic receptors. There are two main types: nicotinic receptors and muscarinic receptors. Now, acetylcholine can't just hang out in the synapse forever or things would get out of control. So an enzyme called acetylcholinesterase, or AC, steps in and rapidly breaks down AC into choline and acetate. This choline can then be recycled back into the presynaptic neuron to make more acetylcholine. The textbook has a nice diagram of this whole cycle in figure 6.3.
Nicotinic and muscarinic receptors, two types of cholinergic receptors. Got it. Let's zoom in on nicotinic receptors first. What makes them unique?
Nicotinic receptors are mainly found in autonomic ganglia, those relay stations, the adrenal medulla, which releases epinephrine and norepinephrine into the bloodstream, and at the neuromuscular junction, where nerves connect with skeletal muscles. In terms of the autonomic system, their presence in the ganglia and adrenal medulla is key. They are ion channel receptors, meaning they have a built-in channel that allows ions to flow through. When acetylcholine binds, it opens this channel, leading to a rapid influx of ions, causing the postsynaptic cell to depolarize, basically changing its electrical potential. Table 6.2 in the textbook breaks down the different subtypes, notably N sub-sub in skeletal muscle and N sub-sub in neurons, including those in the ganglia. So activation of nicotinic receptors generally leads to a fast, almost instantaneous response.
Fast-acting ion channels, okay. Now what about muscarinic receptors?
Muscarinic receptors are a bit different. They're mostly found on those effector organs innervated by postganglionic cholinergic fibers, meaning the target tissues of the parasympathetic nervous system, places like the heart, smooth muscles, and glands. And unlike the direct action of nicotinic receptors, muscarinic receptors are G-protein coupled receptors, which means they trigger a cascade of intracellular events when activated. So instead of directly opening a channel, they initiate a chain reaction inside the cell that ultimately leads to the desired response.
So a more complex chain reaction type of response. Can you give some examples of what happens when different muscarinic receptors are activated?
Sure. For example, activating M sub 2 subreceptors in the heart decreases heart rate and the force of contraction. On the other hand, activating M sub 3 subreceptors, which are found in smooth muscle and glands, causes smooth muscle contraction and increased secretion from those glands. Table 6.2 also gives a breakdown of the different muscarinic receptor subtypes, M sub 1 sub through M sub 5 sub, along with their typical locations and the signaling pathways they activate when acetylcholine binds. So, while nicotinic receptors are like flipping a switch, muscarinic receptors are more like setting off a series of dominoes, a bit slower, but potentially with more diverse and nuanced effects.
That makes sense. So we've covered acetylcholine and its receptors. Now let's move on to norepinephrine, NE, and epinephrine. What roles do they play in the ANS?
Well, norepinephrine is the main neurotransmitter at most sympathetic postganglionic junctions. That means when a sympathetic nerve fiber reaches its target organ, except for those sweat glands, which use acetylcholine, remember, it's usually norepinephrine that's doing the talking. Epinephrine, on the other hand, is primarily released from the adrenal medulla into the bloodstream, acting more like a hormone, though it can have neurotransmitter-like effects in certain brain areas. Both NE and epinephrine belong to a group called catecholamines, and just like acetylcholine, they have their own specific ways of being synthesized, released, and inactivated.
So let's walk through adrenergic transmission then. How are NE and epinephrine made and used by the body?
The journey begins with the amino acid tyrosine. Through a series of enzymatic steps, tyrosine is transformed into L-DOPA, then dopamine, and finally norepinephrine. In the adrenal medulla, there's an extra step where norepinephrine can be converted into epinephrine by the enzyme phenylethanolamine and methyltransferase. These catecholamines are then stored in those same little vesicles within the nerve terminal. This time, a protein called the vesicular monoamine transporter, VMAT, handles the packing. When a nerve impulse reaches the terminal, these vesicles fuse with the membrane and release norepinephrine, or sometimes epinephrine at certain synapses, into the synaptic cleft. Figures 6-4 and 6-5 in the textbook illustrate these synthesis and breakdown pathways really well.
Release the catecholamines. What happens next?
Well, once they're in the synaptic cleft, they diffuse across and bind to their specific receptors, adrenergic receptors. We've got two main types, alpha receptors and beta receptors. Now, to stop their action, NE and epinephrine are mainly removed from the synaptic cleft through reuptake, basically a vacuum cleaner, sucking them back up into the presynaptic neuron. This primarily happens through the norepinephrine transporter, NET. Once back inside, they can either be repackaged into vesicles for later release or broken down by enzymes. The two main enzymes involved are monoamine oxidase, MAO, which is found inside the mitochondria of the neuron, and catechol-amethyltransferase, COMT, which is out in the synaptic cleft and other tissues.
Okay, so we have alpha and beta receptors as the key players in adrenergic transmission. Let's talk about alpha receptors first. What are their general roles?
Alpha receptors are further divided into a sub 1 sub and sub 2 sub subtypes. Generally, a sub 1 subreceptors are typically excitatory and are often involved in causing vasoconstriction, which is the narrowing of blood vessels. This can increase blood pressure. They're also found in other smooth muscle, like in the eye and bladder. Think of I sub 1 subreceptors as being involved in squeezing things, increasing tension. On the other hand, I sub 2 subreceptors are often inhibitory. They can be located presynaptically, acting as a sort of brake on the system by inhibiting further release of norepinephrine. They're also found postsynaptically and have various other effects depending on their location. Table 9-1 in the textbook goes into more detail about the specific subtypes and their effects.
So all sub 1 subreceptors generally constrict or increase tension, while A sub 2 subreceptors often inhibit norepinephrine release.
Got it. Now what about beta receptors? Beta receptors are subdivided into sub 1 sub, sub 2 sub, and sub 3 sub subtypes. Sub 1 subreceptors are predominantly found in the heart. When they're activated, they increase heart rate and the force of heart muscle contraction. Think of them as giving the heart a boost. Sub 2 subreceptors, on the other hand, are mainly located in smooth muscle, and their activation typically leads to relaxation of that muscle. A good example is bronchodilation, widening of the airways in your lungs, which is mediated by sub 2 subreceptors, so they often help to relax things. Sub 3 subreceptors are mostly found in adipose tissue, playing a role in lipolysis, the breakdown of fats for energy. Again, Table 9-1 provides a more detailed overview. So to put it simply, alpha receptors often squeeze or signal, while beta receptors often boost or break down.
Okay, I think we've laid a good foundation here. We've got the ANS with its sympathetic and parasympathetic branches, the chemical signaling using acetylcholine and norepinephrine, acting on their respective receptors, cholinergic and adrenergic. Now, how do drugs fit into all this? How do they actually work to modulate the ANS and produce those therapeutic effects?
This is where the magic of pharmacology happens. Drugs can target practically every step in neurotransmission or directly interact with those autonomic receptors to produce a wide range of effects. We can broadly classify these drugs based on their main mechanisms of action.
Okay, let's start with the drugs that affect the cholinergic system, the system using acetylcholine. What are the main types of cholinergic drugs?
We've got two main categories: cholinergic agonists, which enhance cholinergic activity, and cholinergic antagonists, which block it. Cholinergic agonists, also called cholinomimetics, can be further divided into direct-acting and indirect-acting agents.
So direct-acting cholinergic agonists, those would be drugs that directly stimulate the cholinergic receptors themselves, right?
Yes, exactly. They bind directly to and activate those receptors muscarinic, nicotinic, or sometimes both. Some examples include acetylcholine analogs like carbachol and bethanyl. They behave similarly to acetylcholine but are more resistant to breakdown, so they hang around longer. And then there are naturally occurring alkaloids like pilocarpine. Figure 7.2 and Table 7.2 in the textbook provide details on their chemical structures and pharmacological properties. The main effect of these direct-acting agonists is to mimic parasympathetic stimulation.
Okay, so they're like acetylcholine impersonators, directly activating those receptors. What about the indirect-acting cholinergic agonists? How do they work?
Indirect-acting cholinergic agonists, or cholinesterase inhibitors, work by inhibiting the enzyme acetylcholinesterase, ACE. They basically sabotage the cleanup crew, preventing the rapid breakdown of acetylcholine. This leads to higher levels of ACE hanging around in the synapse, which means a longer and stronger effect.
So instead of directly stimulating the receptor, they're making the body's own acetylcholine more effective.
That's right. Think of it as amplifying the signal. Some examples of reversible cholinesterase inhibitors, meaning they bind to ACE for a limited time, include neostigmine, physostigmine, and edrophonium. Then there are the irreversible inhibitors, like certain organophosphates, such as ecothiopate and the nerve gas sarin. These guys form a super strong bond with ACE, leading to a much longer and more intense increase in cholinergic activity. This can be very dangerous, even fatal, if not controlled. Figure 7.6 and Table 7-4 go into more detail about these different types of inhibitors.
So boosting acetylcholine can be helpful for certain conditions, but too much of a good thing can quickly become dangerous, especially with those irreversible inhibitors. Now let's look at the other side of the equation. What about cholinergic antagonists, those drugs that block the effects of acetylcholine?
Cholinergic antagonists can also be categorized based on which receptor type they target. We have muscarinic antagonists, which selectively block muscarinic receptors, and nicotinic antagonists, which block nicotinic receptors.
Okay, let's start with muscarinic antagonists. What are some examples and what are their effects?
Muscarinic antagonists, also known as antimuscarinics or anticholinergics, include drugs like atropine and scopolamine, which you might have heard of. There's also ipratropium, often used for respiratory conditions. Figure 8.3 and Table 8.1 give you some info on their chemical structures and properties. As you can probably guess, their main effect is to block the actions of acetylcholine at those muscarinic receptors, basically hitting the pause button on parasympathetic activity. This can lead to things like increased heart rate, reduced secretions like saliva and mucus, and relaxation of certain smooth muscles, for instance, in the airways. So they essentially counteract the rest and digest system.
Fascinating. Now, what about the nicotinic antagonists?
These guys block nicotinic receptors. One group is called ganglionic blockers. They specifically target nicotinic receptors in autonomic ganglia, those relay stations we talked about. Examples include hexamethonium and mecamylamine, though they're not used as often nowadays because they affect both the sympathetic and parasympathetic systems, leading to widespread and sometimes unpredictable effects. Figure 8-7 shows their structures. We should also briefly mention neuromuscular blocking agents. These are also nicotinic antagonists, but act specifically at the neuromuscular junction to cause muscle paralysis. Very important in medicine, but for today we're focusing on the autonomic side of things.
Okay, that wraps up the drugs that target the cholinergic system. Let's switch gears to the drugs that affect the adrenergic system. They're norepinephrine and epinephrine pathways. What are the agonists and antagonists here?
Just like with the cholinergic system, we have adrenergic agonists, which stimulate those adrenergic receptors, and adrenergic antagonists, which block them. Adrenergic agonists are sometimes called sympathomimetics because they often mimic the effects of sympathetic nervous system activation. They can be direct acting or indirect acting.
Direct-acting sympathomimetics, so these drugs directly activate those alpha and beta
Yes,
receptors.
exactly. They bind to and activate either alpha receptors, sub-1 sub or sub-2 sub, beta receptors, sub-1 sub, sub-2 sub, or sub-3 sub, or a combination. Examples include norepinephrine itself, epinephrine adrenaline, remember, phenylephrine, which mainly acts on alpha receptors, and isoproterenol, which acts mostly on beta receptors. Figure 9-4 and Table 9-2 in the textbook show their structures and how strongly they bind to different adrenergic receptor subtypes. They generally mirror what happens when the sympathetic nervous system gets activated, but the specific effects depend on which receptors are activated. For example, an on-sub-1 subagonist might cause vasoconstriction, while a sub-1 subagonist would mainly affect the heart.
And what about those indirect-acting sympathomimetics? How do they boost the adrenergic system without directly binding to those receptors?
They're sneaky. They don't directly bind to adrenergic receptors, but they increase the amount of norepinephrine or epinephrine available to activate those receptors. They might do this by increasing their release from those storage vesicles in the presynaptic neuron, or by blocking their reuptake back into the neuron, allowing them to linger in the synapse longer. Think of it as flooding the system with those neurotransmitters. Examples of these indirect acting agonists include amphetamine and cocaine. Figure 9-3 illustrates those different mechanisms.
Okay, now for the antagonists, those adrenergic blockers. What are the main types?
We've got alpha adrenergic blockers, which selectively block alpha receptors, and beta adrenergic blockers, which block beta receptors.
Alpha adrenergic blockers, or alpha blockers for short. What are some examples, and what happens when they block those alpha receptors?
Alpha blockers can be selective for A sub-1 sub receptors, sub-2 sub receptors, or they can block both. Examples of A sub-1 subselective blockers include drugs like phentolamine, prazosin, and terazosin. Figure 10.1 and Table 10.1 show their structures and relative binding affinities. By blocking A sub-1 subreceptors, these drugs can cause vasodilation, the opposite of vasoconstriction, which lowers blood pressure. They can also affect smooth muscle contraction in other organs where A sub-1 subreceptors are found.
And finally, we have the beta adrenergic blockers, or beta blockers, a very common type of medication. What's their story?
Yes, beta blockers are incredibly important and widely prescribed. They can be non-selective, meaning they block both sub-1 sub and sub-2 sub receptors, like propranolol. Some are more selective, like metoprolol, which mainly blocks sub-1 sub receptors in the heart. There are even drugs like labetalol that block both alpha and beta receptors. Figure 10-5 and Table 10-2 provide details on their structures and selectivity. By blocking beta receptors, they can slow down heart rate, lower blood pressure, and have other effects depending on which beta receptor subtype is targeted. Blocking sub-1 subreceptors mainly affects the heart, while blocking sub-2 subreceptors can impact the airways, for example.
It's amazing how we can manipulate this complex system with medications. Bringing it all together, what are the key takeaways about the clinical relevance of all this? Why is understanding autonomic pharmacology so important in medicine?
These drugs we've been talking about are used to treat an incredible variety of conditions. We're talking cardiovascular diseases like hypertension and arrhythmias, respiratory disorders like asthma, eye conditions like glaucoma, even overactive bladder. And this is really just the tip of the iceberg. Each of these clinical applications could easily be its own deep dive.
Right. And the textbook also had those fascinating case studies. Can you remind us of a few examples where knowing about autonomic pharmacology is crucial?
Absolutely. There is a case of the older woman with blurred vision, which could be related to an autonomic dysfunction affecting the muscles in her eye that control focus. Then there was the coworker with possible organophosphate poisoning, illustrating the dangers of those irreversible cholinesterase inhibitors that flood the system with acetylcholine. We also saw the architect with urinary symptoms, where drugs targeting those muscarinic receptors in the bladder could be considered. And there were cases of unexplained high blood pressure, rapid heartbeats, and headaches, all potentially pointing to issues with the sympathetic nervous system, where adrenergic agonists or antagonists might come into play. Even the patient with the pheochromocytoma, a tumor that pumps out excessive amounts of epinephrine and norepinephrine, shows how dramatically the ANS can impact overall health and how managing it pharmacologically is critical.
Wow, it really brings it home seeing how this knowledge applies to real-world situations.
Mm.
So, as we wrap up our deep dive into autonomic pharmacology, what are the most important things to remember?
Well, The main takeaway is that the autonomic nervous system is absolutely essential for keeping those vital involuntary functions running smoothly. It uses chemical messengers, mainly acetylcholine and norepinephrine, to communicate, and these neurotransmitters exert their effects by binding to specific receptors. We've got nicotinic and muscarinic receptors for acetylcholine and alpha and beta receptors for norepinephrine and epinephrine. And of course, drugs can be designed to modify the activity of this system, either mimicking or blocking those neurotransmitters at their receptors or influencing how they are released or broken down.
So by understanding these basic principles of autonomic pharmacology, we can start to appreciate how such a wide range of medications work in the body.
Exactly. You've gained a foundational understanding of this complex system, which will help you better understand how medications interact with your body and how your body regulates itself.
This has been such a fascinating deep dive. It really makes you appreciate all those intricate chemical conversations happening inside us constantly without us even knowing. So, for our final thought today, think about how those seemingly automatic functions are actually the result of incredibly precise and complex chemical interactions. And remember, even a basic understanding of these interactions can open up a deeper understanding of our own health and well-being. Maybe you could even think about some medications you or someone you know takes, and based on what we've talked about, try to figure out how they might be interacting with the autonomic nervous system. If you're feeling extra curious, you can always dive deeper into specific drug classes or clinical applications that caught your interest. Thanks for joining us on this journey.